Right. Last Thursday in a TA meeting, we talked about genetic drift activity for this coming week. We have known how chaotic the activity could be if it is not well organized. I made some revision on the activity spreadsheet, and hopefully it will help the teaching procedure. In this quick video tutorial, I'm going to explain how to use the revised spreadsheet. So here is the spreadsheet. It is organized in a way to help you explain the activity and how to think about genetic drift. So at this point, you should have give your student the worksheet, the data slip, and the coin. And then you pull out this spreadsheet and put it on the board. So based on the spreadsheet, you start to explain to your student that we're going to track the allele frequency change across 10 generations, and we're going to compile the change information for each generation on this spreadsheet. So today, each of us will be a fish, and we're going to start with the genotype of A1, A2, which are two alleles of gene A. So how many students we have today? Let's see, 25. So you put 25 in this cell. You can see the whole line will change because I have made them the same. And then you ask a student, since we all are A1, A2, then how many alleles we have in total? Then it should not be too hard for them to figure out. It is 50. And then you can see this whole line change accordingly. You may also notice some changes occur right here, but don't worry about that for now. And then for the first generation, it's generation 0, and you may ask, okay, now we're all A1, A2 again. Then, since we have a total of 50 alleles, then how many A1 we should have, how many A2 we should have? And yes, of course, we should have 25 A1 and 25 A2. You only need to put A1 here, and then A2 will be automatically calculated for you. Then you tell student, you need to stand up, move around in the classroom until I say stop. And then you randomly pick a partner, flip coin with the partner. You and your partner are going to reproduce 2 offsprings, one for you, one for your partner. And then you're going to determine the genotype of the offspring based on the coin, based on coin flipping. So if you have hat, and that means you give A1. If you had a tail, which means you give A2. So you and your partner are going to flip coin twice. So each of you flip coin twice to determine allele genotype for the offspring. And then they need to write down the new genotype on the data slip. And most important, they only do this current generation. So for this one, that is generation 1. Don't move to generation 2. So if you remember last Thursday, we just kind of keep going on if we are not, we don't understand this. And then you ask a student, now with the new genotype, if you have one A1, raise one hand. If you have two A1, raise both hands. And then you count how many hands in there. Let's see. This time you count 30 hands, which means 30 A1 alleles. And you put 30 right here. And then the A2 alleles will be calculated for you here. And then also the information will be plotted right here in the chart. Now you tell student, now you can move on, stand up, move on, find another partner, for the coin, and then reproduce another new offspring based on your genotype of generation 1. And again, you only do generation 2 for this time. So, and then they do the activity, and then you count how many allele 1 you have. Let's see, for this time, 24. And you can see, yeah, again, number will be changed again for you. Then you can just keep doing this all the way down. Right? And at the same time, you can see the chart will be updated at the same time for you. Then when you finish the 10 generations, you leave this spreadsheet on the board, and then your students are going to repeat this activity within their table group. So that's it.